Hi dear students, welcome back to one more session of genetic engineering. Today we are going to talk about the methods to detect recombinants. In the previous sessions we were finding different steps of genetic engineering. What are the methods to transfer the gene of interest or the recombinant DNA onto the host cells? And now we are going to explore the methods which have been used to find out the recombinants. Now, why do we have to screen recombinants? After the introduction of recombinant DNA into the host cells, it is very essential to identify those cells which have received the recombinant DNA molecule. And that is mainly referred to as a screening or the selection of the recombinants. We can see that the vector or the foreign DNA will express in the recombinant cell and certain characteristics or traits would be present in the recombinant cells when the vector or the foreign DNA is being present in the recombinant cell. But those cells which are being found to be non-recombinant or which do not contain the foreign DNA will not express those traits. So those methods, we employ certain methods here to find out whether the host cells are become recombinant or not. All those techniques mainly are the ones which we are discussing here today. Now, the first technique what I would like to speak is on the insertional inactivation. Insertional inactivation is the inactivation of a gene upon the insertion of another gene in its place or within its coding sequences. And this could help in the selection of recombinant colonies in our DNA technology. Now, when you hear this like this, you might find it complicated. But just have a look at this vector. What is this vector? This is a plasmid PBR322. And to produce a recombinant DNA, we know that every plasmid which we use is being cut using restriction endonucleases. And a vector is always being marked by the presence of selectable markers. Now, in the case of PBR322, we know the ampicillin resistance gene as well as the tetracycline resistance genes are two antibiotic resistance coding genes and they can be used as selectable markers. What do you mean by selectable markers? Selectable markers are those markers which will enable us to know whether this particular vector is present in that particular host cell. So, if you take a host cell and if you insert this particular plasmid into it, that particular host cell will gain two important characters. One is ampicillin resistance and tetracycline resistance. So, both the resistance of ampicillin and tetracycline is possible when the whole vector, which is not being cut by any enzyme, is being inserted into the host cell. Now, as we know in our DNA technology, we take the vector and we have to cut it using a restriction endonuclease and we put a gene of interest. So, consider a case in which you have cut this vector using an enzyme restriction endonuclease BAM-H1. I'm cut here at this particular region and I have put a gene of interest in this particular region. So what happens when if my gene of interest is being put into the BAM-H1 site because I've cut it, I have taken my gene of interest as well as I've cut this plasmid using a BAM-H1. So there is a cut over here and my gene of interest is put over here. So what happens? When the gene of interest is being put over in the BAM-H1 site, which also codes for tetracycline resistance, what happens? The inactivation of the tetracycline resistance gene will happen. Okay. So depending upon which location you insert your gene, the marker, the character or the trait which is being coded by the marker gene will be inactivated. And that can be used to find a the recombinant clones. Now, instead of the tetracycline resistance, if I'm cutting it with PST1 and putting my gene of interest here, the ampicillin resistance gene will be inactivated. So, the process of insertional inactivation is the inactivation of the gene upon the insertion of another gene. So, here in this particular case, if you cut tetracycline resistance, this vector using a BAM-H1, and at the same time, if you put a gene of interest here, what happens? This resistance gene will be inactivated. Now, using that technique, you can find out whether a particular vector is or a recombinant is being formed. And let me just tell you a basic thing. If you want to identify if a plasmid has been obtained or a recombinant plasmid or a plasmid is generally entered into a host cell. If you consider a host cell and if it has been what transformed with a particular plasmid, the host cell will also get the character coded by the particular plasmid. So, if you're putting PBr322 as such, that is without cutting at tetracycline or at ampicillin resistance gene region, if you just take the whole vector and put it into an E. coli cell, what happens? The E. coli cell will get the resistance for both ampicillin as well as what? Tetracycline. So, if you grow the cell or the E. coli cells on the media with the antibiotic, what happens? There would be more cells, there would be cells which are having resistance to that antibiotic. Now, there would be, see, you are finding here some cells, tiny cells over here. These are not being considered to be antibiotic resistance and they are not being grown here. So, like that, to check the presence of E. coli cells which are being transformed with a particular plasmid, you can just grow the cells on a media containing the correct antibiotic. Now let's go into insertional inactivation. Consider a case as we had mentioned earlier, that is we are inactivating or we are cutting this particular vector at the tetracycline resistance gene and then your gene of interest is put here. So now in such cases, you can have different types of colonies. If you get non-transformed cells, what do you mean by non-transformed cells? Those cells which do not have the entire plasmid. Those cells will not grow on ampicillin as well as tetracycline containing media. So if you put those cells and put it on a media containing ampicillin and tetracycline, okay, these non-transformed cells which do not contain the plasmid will not grow over here. And if you're having transformed cells, that is, they are transformed colonies which can grow in ampicillin as well as tetracycline media. That means it is containing the particular plasmid. And sometimes you can see that even if we cut it at the tetracycline resistance gene portion, sometimes the vector might recircle and the gene of interest might not enter into it. That means it is transformed with a non-recombinant or an unaltered vector. So in such case also, it can grow in both ampicillin and tetracycline resistance. Then finally, you have another case in which the case which we are searching, that is the transformed with a recombinant vector. That is, your gene of interest is being present. Now, if it has been put in the tetracycline resistance gene portion, as I had mentioned earlier, it will be capable of growing in a media containing ampicillin but not on tetracycline because the gene of interest has been put in the tetracycline resistance. See, I've cut it over here and I've put my gene of interest. So, the recombinant clone will lose the tetracycline resistance process ability, but it will retain the ampicillin resistance. So, The transformed with recombinant vector, those colonies which are transformed with the recombinant vector, will be capable of growing on ampicillin media but cannot grow on tetracycline media due to insertional inactivation. And thereby, we can detect the recombinant clones easily from the master plate. Now, what do you think? Like, this is a master plate, and you will blot all these from the master plate. You will blot them onto the different medias. Okay, and then you will identify which are the colonies. So you compare the each media with the master plate and find out where is the particular colony and what is its character. I guess you got an idea about antibiotic screening as well as insertional inactivation, and this method is being used for the determination of recombinants. Now, you cannot generalize that what ampicillin resistance and tetracycline resistance. It is, I'm talking about ampicillin and tetracycline because of this example PBR 322 which I have taken over here. Based upon which vector you are using, based upon that, the screening strategy or the media which you use or the antibiotic which you use for the screening would change. So this is the first method for the screening of recombinants, the insertional inactivation technique. So we will continue in the next session. Thank you for now.